Good evening, sir.
Good evening, sir.
Good evening, everyone.
Good evening, sir.
Yeah, good evening, everyone. Just in 1 minute, we are starting.
Sir, we have bio today, right?
Yes. Yeah, we are bio.
Sir, I got my science results and I got 30. Actually, I did not see one question of three marks, so I didn't attempt that.
Which one?
So, one biology question was there. I knew the answer, but I forgot to write the answer of 3 marks.
Okay. So,
I would get 33.
okay, anyways, let's continue now. So
Okay.
today we'll continue with bio. And in bio, we had started with cell. We were doing the structure of cell. We had done the animal cell and the plant cell. We are doing out the differences. And can you name me some of the organelles which are present in plant cell but not in animal cell?
So
So,
cell wall.
wall, chloroplast,
Sir,
Yeah,
mitochondria.
cell wall,
cell wall,
chloroplast,
chloroplast.
mitochondria,
So,
Sir, ribosomes.
Sir,
cell wall,
mitochondria and ribosomes are common in both the animal and plant cell. Sir, are you asking the organelles
okay.
which are different
Name
from
some organelles.
animal cell?
No, organelles which are present in plant cell but not in animal cell.
Sir, means those organelles which are differently present in plant cell.
Plastids, large vacuole.
Yes, a large vacuole, very good.
Sir,
And
plastids.
plastids.
Sir,
And
but I know I forgot the term, but there is one type of organelle which give colors to fruits, fruit shells. What is that?
organelles which?
Give colors like if we take red and apple,
Okay.
some organelle gives the color.
They are chromoplast.
Okay, sir. Chloroplast,
Chromoplast.
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 Sir, chloro, chromo, and
Another is
Leuco,
also there, which gives white color.
leucoplast.
Yeah.
You have chloroplast, chromoplast, and leucoplast.
yes, sir.
The leucoplasts are the ones.
Sir, is lysosomes present only in animal cells? I
No,
mean
they are
plant cells, sir. Lysosomes are in plant cells, right?
both. They are present in both plant cell and animal cell.
Sir, why are they called as suicidal wax, sir? Lysosomes?
Lysosomes, they contain digestive enzymes.
So can I tell?
Yes.
So lysosomes contain digestive enzymes. If there are no bacteria or anything, So the lysosomes produces the enzymes that can consume the cell.
So lysosomes they have digestive enzymes and they can digest even the contents of cell.
Sir, but this happens only when there is a disturbance in cellular metabolism, no, sir?
When there is
Sir, when there is a disturbance in the cellular metabolism only it will happen, no, sir?
uh, then also it can happen or if by the bursting of the lysosomes, even if the lysosomes burst out, then also it will happen.
Sir, how does cancer occur?
Okay. So let us see first of all what is cancer.
Sir, cancer is the rapid dividing of cells.
Yeah. So cancer is the rapid division of the cells of a similar type, unwanted rapid division of the cells. Right? So if somewhere this happens in our body, so we call that condition to be the cancer. The cells are said to be the cancerous cell. Okay. And why it happens? Obviously, there is some difference in metabolism that occurs that may lead to cancer. The
Sir,
regions of cancer, they are still unknown that how the cancer begins. But yes, it's known that how it happens. It like
sir, and how is there any cure for any type of cancer yet?
uh, is there any?
Cure for cancer, any type of cancer.
Yeah, of course, there are different cures for cancer.
Sir, what is
For
it?
example, there are
And
there are there are chemical methods, surgical methods, and radiation methods.
sir, can you explain
Surg
this?
huh? Yeah,
Can you
during the
explain?
during chemical methods are given as medicines so that those medicines can kill the cells. Okay. So they not only affect the cancerous cell, they affect even the cells of our body.
So means the chemical method is a bit harmful to us.
All the methods are harmful. So that is why whenever you see that the chemical methods are adopted for the patients, right? So you generally observe that the patients who undergo for the treatment of cancer, they lose their hair. Right. So the loss of hair that people suffer over there, that is all because of those chemo treatment that is going on. Because of the chemicals which affect their body also. Their body is not in a situation to even handle their body. It's not in a situation to even handle their body. Some people are not even able to stand up. It makes them so weak. The chemotherapy makes them so weak that they become almost uh,
Sir, can you spell the name of the therapy?
chemotherapy. I'll just give you just 1 minute. I'm just trying to open up the PPT of cell.
Okay, sir.
Just 1 minute. Oops. So PPT of cell. Just 1 minute.
Sir, means this chemical method can even make the people paralyzed.
Yeah, it can. It can. Okay, anyways, currently the PPT is not available. I will search for it later. Let's continue without.
Sir, I have a doubt.
Yeah, what happened?
Sir, actually, we know that every material in the earth is made up of any element. What are cells made up of?
Okay, see, if you look up at the chemical composition of the cells, so cells are made up of different chemicals. Like in them, the most ones is your proteins. Okay. Now, if you look up for yes, what elements the proteins are made up of, so proteins are they are made up of amino acids. These amino acids they are made up of they are made up of nitrogen, hydrogen, oxygen, and carbon. So these elements they make up the amino acids. Those amino acids they make up the protein. Okay. Even after amino acid they make polypeptides. Then polypeptides they make the proteins. Okay. So the main constituent of the cell is the proteins. Then of course you have the fats, which are the oily parts. You have the fats. Then you have other uh, sugar parts, that is your carbohydrates. So these are the all things of which a cell is made up. If you are thinking of the living component of the cell, that is necessarily the proteins or the amino acids. Got it?
Sir,
Sir,
Yes.
what is
Yes.
sir? What elements? Can you? What is CN?
Cn, it's not Cn. I've done nitrogen, hydrogen, oxygen, and carbon. These are
Okay,
the
sir.
building blocks.
Okay, sir.
Okay. Okay, let's continue. Today we'll discuss about plant cell in detail. So in plant cell, as you told that there are three types of plastids. Plant cells have a special organelle called plastids. And the plastids of plant cell, they are of three types. These plastids, they are either chloroplast, chromoplast, or leucoplast. Okay. So everything I'm clarifying, chloroplast, chromoplast, and leucoplast. Chloroplasts, they contain green pigment called chlorophyll. The chromoplast, they contain other coloring pigments, other color pigments. Since chlorophyll is the green pigment, it is contained in the chlorophyll only. Then chromoplasts, they contain other color pigments, right? Like the red color pigments. You observe that apple is red in color. You observe other tomato which is red in color. You observe other things, mango being green in color. So all these colors they originate from the chromoplasts. Then you have the leucoplasts. So the leucoplasts they are colorless. Leucoplasts are colorless, and so leucoplasts they are colorless and they store generally starch. Now, starch is a form of food that is present in the plant cells. In human body, it is present in the form of glycogen. In plants, it is present in the form of starch. So, starch can be broken down further to carbohydrates. Okay, the starch is a carbohydrate only. So, starch can be further broken down to glucose and maltose. Got it? So, I have understood the structure of plastids.
Sir, what
Now,
is starch
today,
composed of?
what is starch? Starch is a form of carbohydrate.
What is it composed of?
Okay. See, a starch, it's composed of glucose and fructose. You have glucose and fructose, or glucose and fructose, they are combined to form maltose first. Okay. These are the sugar molecules, they combine to form the maltose, and the maltose further combine to form the starch. So you can say your starch is made up of glucose and fructose. Okay, it is a polysaccharide. Polysaccharide
What
means
is
it's
fructose?
made of, yes,
What is fructose? FRUC.
fructose. Okay, fructose is the sweetening element found in fruits.
So can we tell it's composed of sugar?
Yeah, if you look up at the formula of sugar and glucose, so glucose is C6H12O6. Fructose is also C6H12. Okay, but still they differ in their molecular compositions. Okay, they have their formulas similar. A little bit of differences are there. And your maltose, it consists of both glucose and fructose combined together. Okay, so if two molecules of glucose combine, it gives rise to a sucrose molecule. If just two molecules of glucose combine, it gives a sucrose, that is C12H22O11. That is a sugar molecule.
Sir, means fructose and glucose both are sugar only. Means
Yeah, both. Yes,
okay, sir.
yes. Okay. So both glucose and fructose, they are sugar only, but glucose is the simplest form of the sugar. Fructose is also the simplest form of the sugar. With the difference between them, fructose also has some fruity smells. Okay. So glucose and fructose, they are both sugars. They combine to form the maltose. Maltose is also a type of sugar. The sugar that we eat is C12H22O11. It's also called sucrose.
Sir, what are
Different molecules.
sir,
Yes,
you told sir, that fructose is composed of some thing other
something. No, no,
no,
no.
sir. You told other than sugar, fructose is composed of something else.
Not something else. The molecular structure is this one only. It has 6 atoms of carbon, 12 atoms of hydrogen, and 6 atoms of oxygen. But their way of arrangement, the way the molecules or atoms are arranged, is different from that which are arranged in glucose.
Okay, sir. The molecular
If the,
arrangement is different in both of them.
yeah, the molecular arrangement is different, but they are the same. So glucose and fructose combine to form the maltose. Then maltose combines to form the starch, which is a polysaccharide. Okay. Generally, the polysaccharides, they are not sweet in taste, but once they get converted to maltose or sucrose, they become sweet in taste. Like if you start chewing your chapati, so initially it will not taste anything, but as you continue chewing, so within a minute it will start tasting sweet. So that is, it starts tasting sweet because of formation of maltose. We are eating starch. When we are eating a chapati, or eating starch, so that starch is getting converted to maltose, which starts taking sweet. It becomes sweet in it.
Sir, it turns into maltose because of the saliva present in our mouth.
Yeah, because of the saliva present, it converts into maltose. Okay, and that maltose is further converted to glucose and fructose after digestion process is complete. So this was about the plant cell. Now we'll study about osmosis and diffusion, the two important cell processes. So the process of osmosis and diffusion, let's say we have
Sir,
earlier heard about the process of diffusion. Yes,
sir, can I tell?
yes, what is diffusion?
Sir, when the particles move from a higher concentration to when the particles of water move from a higher concentration to lower concentration.
Particles of water. I'm
No,
talking
sir, sorry.
diffusion.
Movement of higher movement of particles from higher region from low to the lower region.
Sir, the movement of particles from a higher concentration region to the lower concentration region is known as diffusion.
Okay, so movement of solute particles from a region of high concentration from a region of high concentration to a region of low concentration. The movement of solute particles from a region of high concentration to a region of low concentration is called diffusion. So during the process of diffusion, we observe that the solute particle, they move from a region of high concentration to a region of low concentration. Can you give any examples of this?
Sir, if we spray any perfume in the air, we can smell it at another, means we can smell it after some meters also.
Yes, if you just spray perfume in a room, so in that room the concentration of perfume is high at a certain corner, then it diffuses to everywhere. It diffuses to all the place of the room. You can smell it everywhere. So the particles of the perfume, they move everywhere. Okay. So they are
Sir,
moving. Yes.
sir, can we add in the diffusion point until equilibrium is reached?
Yeah. Even when the equilibrium is reached, the movement of the particles continues. They continue moving throughout since the particles are continuously moving due to their temperatures. So the particles keep on moving, but their concentrations remain the same. So at the microscopic level, the particles are still moving. They are not stationary. Okay. So diffusion, it is just like if you light an agarbatti or incense stick in your room, so you'll be able to smell it everywhere. It can be something like this. Suppose if you keep a beaker, and in this beaker you put a drop of ink at a place. Let's say I put a drop of ink over here, and I fill this beaker with water. So this ink will spread everywhere in the beaker. Since at this corner it has a high concentration, but it will spread everywhere since all the regions they have low concentration. Okay. So this ink particle will spread everywhere and it will make the complete particle blue, complete water to be blue. So what has happened here? Diffusion of ink has taken place from one corner to every place in this beaker. Okay. Everyone is clear about the diffusion?
Yes, sir.
Okay. So which particles are moving in the diffusion process? Solute particles or solvent particles?
Solute particles.
Solute particles. From where they are moving?
From the higher
Higher
concentration to lower
concentration to lower concentration.
From higher concentration to lower concentration. So from whose higher concentration? From the higher concentration of solute or from the higher concentration of solvent?
higher concentration of the solute.
Yes. So see, these points have to be clear before we move on to osmosis, that we are considering here the concentration of solute particles. It is the high concentration of solute particles, and the particles are moving from a region of high concentration to a region of low concentration. Okay. Now, another process which is similar to diffusion, but over there in that process, there is no movement of the solute particles. We restrict the movement of the solute particles and allow the movement of solvent particles. And this process is called osmosis. So osmosis, it is the movement of solvent particles it is the movement of solvent particles from region of low concentration of solute to high concentration through a semi-permeable membrane. So through a semi-permeable membrane, there is movement of solute particles. See, in the osmosis, there is not movement of solvent particles. The solvent particles are not moving. We have restricted the movement of solvent particles by putting a semi-permeable membrane between the two solutions.
Sir, also the cell wall.
Yeah, it can be also the cell wall. So the semi-permeable membrane, let us see how does osmosis occur and what we are talking of osmosis. So here we contain two containers. So let's say we have here two containers, and the two containers, they are containing liquids. So both the containers, they contain solutes. Okay. And they contain some amount of solvent also. So let them contain some amount of solvent. And let's say these are some solute particles. And here you have a lot of solute particles. So clearly I can see, if I compare the two sides, let this side be your side A and let this be the side B. So the side B, it has the concentration of side B is high. It can clearly be observed that on the side B, the solute has a high concentration, and on the side A, the solute has a low concentration. Right? Yes. If you observe, and in between, we have put a semi-permeable membrane. The role of the semi-permeable membrane is stop the movement of the solute particles. The semi-permeable membrane will not allow the solute particles to get mixed up. Now, due to the concentration gradient, what will happen? To maintain the concentration, now the solvent particles will start moving. That is, the molecules of water, they will start moving through it. Got it? So here, if you have these water on this side, we have the low concentration of solute, and on this side I have the high concentration of solute. So if on side B more particles of water are added, then the concentration would become the same. Is this understood to everyone? That if from side A some particles, they start moving to side B, which particles are moving? The particles of water are moving. So if the particles of water start moving from side A to side B, so the concentration of the solute particle on side A will increase, and the concentration of solute particles on the side B will decrease, and hence a concentration gradient that was there, it will be removed. The two solutions would become of the same concentration. Okay. So this phenomena is called as osmosis. Here we observe the movement of water particles.
So we can see this in real life while if we see anyone cooking vegetables and if they add salt, we can see water coming out of it.
Water coming out of what?
Out of the vegetables.
Yes, water coming out of the vegetables, that is of course your osmosis. In the similar way, you can see resins. If you soak some resins in water, so we observe that the
They often
water,
break.
yeah, they become swell up. The resins, they become swell up. They absorb water. Okay. So the absorption of water is over there by the process of osmosis.
Sir, but I have a doubt in that process. In the resins, there is no liquid, there is no amount of water, so there is low concentration of water inside and higher concentration of water outside it.
So, so
Why not it is diffusion?
yeah, first of all, it is a semi-permeable membrane. Right? There is a semi-permeable membrane. Also, there is no movement of solute particles. There is movement of solvent particle only. It is water particles which are moving inside, and not the particles. See, inside here you have high concentration, and here you have low concentration of what? Here you have high concentration of sucrose and other substances, and outside their concentration is low. Okay, so when it is added in water, it should be like whatever the sugar and all is inside this, it should be able to move out. But since because of the semi-permeable membrane, it is not moving out. Okay, since it doesn't move out, therefore to maintain the concentration of water from the outside starts moving inside. Okay, so hence it is the process of osmosis and not diffusion. In diffusion process, it is the movement of solute particles, but in the osmosis process, it is the movement of solvent particles. Okay, so here we observe the movement of water particles from outside into the regions. Anything else?
Okay, sir.
So
No
everyone
doubt,
has
sir.
understood. Yes, everyone has understood about osmosis and diffusion.
Yes, sir.
Okay, so see, in the process of osmosis, a semi-permeable membrane is required. It is a must. Without a semi-permeable membrane, there will not be any transfer of substances. Okay, without semi-permeable membrane, there will not be any osmosis occurring. But in the diffusion process, whenever diffusion is occurring, so in the diffusion process, yes, there will be. In the diffusion process, there will it can occur even without a semi-permeable membrane. No membrane is required for the diffusion process. In the diffusion process, it is the movement of solute particles, whereas in the osmosis process, it is the movement of solvent particles. Got it? In some books, you may find osmosis to be defined something like this. It is the movement of solvent particles from the, or it is the movement of water from its region of high concentration to low concentration. Okay, but when they are writing, they are referring to the concentration of water and not the concentration of the solute. Okay, so now it's clear about osmosis and diffusion.
Yes, sir.
Okay, now see a similar process that we use every day in our life, that is reverse osmosis. Anyone has heard about reverse osmosis?
So means the movement of solvent particles from higher to lower.
Okay, reverse osmosis. Where do you see in your life?
So water purifiers.
Yes, water purifiers. They use the process of reverse osmosis, and hence the devices they are known as RO. RO means reverse osmosis. Okay. So the device is called as reverse osmosis or RO.
So how?
See, osmosis can naturally occur in this way. Like here we observe that water is moving from side A to side B. Okay, here the concentration of the water particles is high, so it's moving to side B. We try to use some energy, generally the electrical energy. So with the help of the energy, we try to move the water particles from the side B to side A, and restricting the movement of the solute particles. So what will happen? The concentration of water molecules on the side A will increase, right? And
Yes.
hence we will say here that the water is becoming pure. So this is how your reverse osmosis occurs, or RO devices work.
So means the filter purifier doesn't filter the water 100%. There will be few bacteria or something present in that.
Yes, there will be. Okay, and whatever they use the two chambers over here. So one of the chambers, this will be for the impure water, and this will be for the pure water. And now here, from the impure water particles only will be allowed to move by the use of energy. Okay, so from the impure water to the side of the pure water, all the water molecules will move. Now, depending on the permeability of the membrane, there will be movement of certain salt particles. Got it?
Sir, actually, in my school, my biology sir told about one hypotonic solution, which means
Okay, so
yeah,
hmm.
a hypotonic solution. Sir, do you know about that?
Okay, so just we are discussing about that our next topic is the same tonicity of the solutions.
Okay, sir.
Okay, we call them as the tonicity of solution. Tonicity means which one is hypotonic, which one is hypertonic, and isotonic.
Sir, so the this about the resin, if we keep it in water, it's swellens up. So doesn't it come under hypotonic solution?
Just first of all, see what is hypo, hyper, and iso.
Okay, sir.
This concept we call as the tonicity of a solution. Okay, so the tonicity of a solution, it means the solution may be either hypotonic, hypertonic, or isotonic solution. So what does it mean by tonicity? So suppose I put a cell content, a cell in three beakers with three types of with solutions of three levels. Let's say here you have one of the beaker. Okay, so here we take three beakers, and in all the three beakers we add a cell. So suppose here we add a cell inside it. In each of them I am adding a cell, and let us name them. Let's say the first beaker is your beaker A, this is beaker B, and this is beaker C. Okay. So suppose in the beaker A, the concentration of both of them, the concentration inside and the concentration outside is equal. So here both the concentrations are equal. In this one, suppose inside we have high concentration and here we have low concentration. Suppose here it's inverse. Here you have low concentration inside and a high concentration outside. Okay. As we know that a cell membrane is a semi-permeable membrane and it allows the movement of certain particles and certain particles are not allowed. Okay. So here the cell will not allow the movement of the particles from inside to outside, but surely it can allow the movement of water particles. In the first case, the two have the same concentration. So there will not be any movement of the substances from inside to outside or outside to inside. Even if there is any movement, so it will be maintained. Okay. So such a solution where the concentration of the cell and the concentration of the solution is same, we call such a solution to be an isotonic solution. Okay. Iso means same. Now here, the concentration of solution is low. I have a low concentration solution, and in the low concentration solution I have put a cell. So this one is a hypotonic solution. Hypo means low. The concentration of the solution is lower than the concentration of the cell. Here this is a hypertonic solution. Hypertonic means the concentration of the solution is high. Here solution has high concentration. Solution concentration is high and the concentration of the cell is so this is a hypertonic solution. Now see what are the results expected in this. So in this hypotonic solution, since the concentration of the cell is high, okay, so it's just like your resin soaked in water. So to maintain the concentration, as we know that osmosis occurs from a region of low concentration to high concentration. So here there will be movement of water particles from outside to inside of the cell. Am I correct over here? Yes.
Yes.
Everyone has understood this, that in the second case there will be movement of water particles from outside to the inside of the cell. Okay, so this movement of water particles from outside to the inside of the cell it happens, and here we observe that osmosis is occurring. Osmosis is occurring from outside to the inside. And now what effect will it have on the cell? Suppose we had put a plant cell. So inside the plant cell, you have the cell wall, and within the cell wall, you have a cell membrane, right?
Sir,
Then
this
you
cell
have
will shrink. Sir, it will shrink in the
no,
hypotonic solution.
no, it won't shrink. It's here the solution is hypotonic. It has low concentration. So water from outside is moving inside the cell. When more and more water molecules move inside the cell, so it will push the cell membrane towards the cell wall. As a result, your cell will become turgid. It will become hard. Okay. But if you put up the same cell inside a hypertonic solution, here the concentration is outside is high, but the concentration inside the cell is low. When inside the cell the concentration is low, so now the water particles will start moving from inside of the cell to the outside. Since osmosis occurs from low concentration to high concentration. So here the osmosis occurs from low concentration to high concentration, and hence here in this case, I will observe the movement of water particles from inside the cell to the outside the cell. As a result, so in this case, what is going to happen? In this case, my plasma membrane will become flaccid. It will shrink. Here, plasma membrane or cell membrane, it will shrink from all the sides. Right? Since its contents are decreasing. So in the hypertonic solution, a cell membrane shrinks, whereas in a hypotonic solution, the cell membrane will swell up. Got it? Yes. Anyone having any doubts with this?
Sir,
Hypotonic,
hypotonic
hypertonic.
No
solution.
doubt, sir.
Sir,
Yes,
sir,
hypotonic is.
a hypotonic solution, the cell will become rigid.
Yeah, in the hypotonic solution, the cell will become rigid. Okay. As osmosis will occur from outside of the cell to the inside. There will be movement of water particles from outside to the inside. And depending on this, we have seen two processes. We call it plasmolysis and deplasmolysis. So here what is happening? The plasma membrane is getting pushed towards the cell wall. It is causing more and more pressure. This process is called as, not this one. Here, when we put the cell inside a hypertonic solution, there is movement of water from outside, from inside to the outside, and hence the cell membrane became flaccid. So this situation is also called as plasmolysis. So let us try to define what is plasmolysis. Okay. So plasmolysis, here, when a plant cell is placed in a hypertonic solution, so when a plant cell is placed in a hypertonic solution, water from it moves out, causing plasma membrane to shrink. So here there is shrinking of the plasma membrane. When a plant cell is placed in a hypertonic solution, water from it moves out, causing the plasma membrane to shrink. So your plasma membrane shrinks over here. This process is called plasmolysis. So I'll show you plasmolyzed cells. So understood what happens in the case of plasmolysis. In the case of plasmolysis, the water from the cell moves out, okay, causing the plasma membrane to shrink. And this type is called as plasmolysis. So let us say a plasmolyzed cell. I'll show you just a plasmolyzed cell.
Sir, plasmolized cell means the cell in which osmosis is occurring.
Mm, the cell in which plasmolysis has occurred and the plasma membrane has shrunken. So see here, this is a plasmolyzed cell. This one you can observe. Here also you can see how the plasmolysis has occurred. See, earlier it was fully towards the cell membrane. The complete plasma membrane was uh, towards the cell membrane. Now here in the second case, we observe that water is moving out. So when water moves out, then the plasma membrane starts leaving the cell wall. It starts shrinking. So see here, the plasma membrane shrinks, it becomes small. So this situation is called as plasmolysis.
Sir,
And we
means
obtain
the sir, it means the cell inside the cell wall shrinks, but the cell wall remains like that.
no, cell wall doesn't shrink, cell membrane shrinks.
Oh, okay, sir. I'm also telling the same thing. Cell membrane and all organelles also shrink, but the cell wall remains the same.
Yeah, cell wall remains as it is. As cell wall is rigid, it is hard, it cannot shrink. Okay. So cell wall is freely permeable. It allows almost all the particles to enter the cell.
Sir,
This type
means
of
what are the pores called which cell wall has?
cell wall is just a molecule. It's a cellulose molecule.
Sir, are there
Cell
any
wall.
pores? Are there
Cell
any
wall
pores
is
in the cell wall which allows the movement of particles?
no, they are not pores. It is a that is why it is called as a cell wall. There are no pores over there. But now if you're thinking how the particles are moving, how the water is moving, so as the at the microscopic level, the bonding of the molecules, the bonding of the cellulose molecule is not that there are uh, there are already spaces in the molecules. So through that there is movement of water particles. But
Okay.
your cell membrane has a typical structure which can allow certain particles to pass through it and certain particles not. Got it? So here you have this one is a plasmolyzed cell. It is plasmolyzed cell. It is because of the movement of water from inside the cell to the outside, and this happens in the hypertonic solution. So see the same thing we had done about the hypertonic solution.
Sir, means the vacuole is only getting less or big.
See, even the vacuole also becomes small. The cell membrane also shrinks.
So means it is dependent upon the vacuole.
The size of the size of the vacuole only depends upon all these things. Okay. So the size of the vacuole, it depends on the amount of water present in the inside the cell. Okay. So see the first one, it is hypertonic. The second one, isotonic, and the last one, it occurs in the case of hypotonic solution where the cell becomes rigid. See here, the plasma membrane is pushed towards the cell wall and it is causing or creating a pressure on the cell. Anyone having any doubts in this?
No doubt, sir.
Okay. So I think everyone has understood today the diffusion, osmosis, plasmolysis, and deplasmolysis. What is deplasmolysis? Can anyone tell what is deplasmolysis? See, deplasmolysis is the opposite of plasmolysis. Like in the hypertonic solution, the cell becomes plasmolysed. And if you keep this plasmolysed cell in a hypotonic solution, it will again regain its shape and it will become turgid. So this process is called as deplasmolysis. If you put a plasmolysed cell in a hypotonic solution, it again becomes the same thing. So that we call as the deplasmolysed. Okay. Okay, so bye all of you. See you again in next class.
Thank you, sir.
Bye, sir. Thank you.
Bye, sir. Thank you, sir.